the two functional groups, the one that defines the activity of the compound. First is the carbonyl oxygen. The carbonyl oxygen is your carboxyl group or a ketone group. And the other one is the hydroxyl group, the OH group. Okay? So these are the two reactive groups, or what you call functional groups of your molecule. Okay? So carbohydrates, you take it in, you put it in your mouth, you chew on it, and it is partially digested by your salivary enzyme, which is amylase. Okay? The longer it stays in the mouth, the better is the digestion. No? Okay. Then it goes down into your stomach. In the stomach, there's no enzyme for carbohydrate. The salivary amylase that goes with the food that is going into the stomach is made or rendered non-functional because your salivary amylase does not work in an acid pH. And you know how your stomach is. It's so acidic. And it has usually a pH of around 1 or 2. So it's quite acidic. Then, although there is some sort of what you call this degradation of the carbohydrate molecule by your acid, the hydrochloric acid, and we call it degradation by your hydrochloric acid, or degradation. And then it goes down to your first part of the intestine, and that's your duodenum. There in the duodenum, you will have secretion from the pancreas, which is known as the common bile duct, because the pancreatic duct and the hepatic duct from the liver joins to form a common duct that empties into the duodenum. And so there you have your digestive enzyme for carbohydrate, the pancreatic amylase. Pancreatic amylase and salivary amylase are similar. They only differ in their location. So their product of digestion by this enzyme is up to maltose. Okay? Up to maltose. Now, as the food travels downward to the other parts of the small intestine, there on the intestinal walls or intestinal mucosa or epithelium, intestinal epithelium, are cells that produces enzymes, intestinal enzymes for carbohydrates. and they usually act on the disaccharides already that are being formed. And the foremost disaccharide is maltose. Okay? Now, disaccharides coming from your diet, like your sugar, sucrose, no? Your sucrose is not acted upon by your salivary amylase nor your pancreatic amylase. You have an enzyme, your sucrase, which acts on it coming from the intestinal epithelium. So you have the enzymes from your intestinal epithelium, which acts on the disaccharides. Collectively, they are known as disaccharide. Now, for maltose, you have maltase. No? For sucrose, you have your sucrase. No? That acts on it. And sugar coming from milk, which is lactose, is acted upon by intestinal enzyme lactase. Okay? So as a result, you will have monosaccharides. And the predominant monosaccharide is glucose. Because maltose is made up of 2 molecules of glucose. Your sucrose is made up of a molecule of glucose and fructose. And your lactose is made up of a molecule of glucose and galactose. So the predominant sugar or monosaccharide is glucose. So the major product of digestion of carbohydrate is glucose. However, the 2 other monosaccharides that are also high in amount, but not as high as the amount of glucose, your fructose and galactose, should remember that they are isomers of glucose, which can easily become glucose through the action of your isomerases. So most or the largest amount of monosaccharide that goes into the blood is your glucose. Although there might be fructose, there might be galactose, but they are in small amount. Unless, of course, you come up with a high fructose syrup, which now predominates in your the sweetening agent of your favorite drinks, then you will have a predominance of fructose. Okay. So this is, of course, seen in Western diets. Western diets. And we Filipinos are copycats. We just follow. Especially American diet, we usually follow what the Americans are eating. We consume their products, in which case, of course, our own food, prepared food from your grandmother down to your immediate parents, you will have more nutritious food compared to this copied diet, Western diets. No? Okay. So that's why what we have now is a pandemic, which is, call it COVID-19 pandemic. Actually, there is another pandemic going on silently. And that is, we have several. That is not only that what I am trying to refer to is diabetes mellitus. There's another pandemic, obesity. There's another pandemic. But we are not alarmed because they will not kill you right away. No? So we are more alarmed with this kind of pandemic that kills us in a short time. But diabetes mellitus, you can still go on. Or obesity. No? Now, together with obesity and diabetes mellitus, you will have atherosclerosis, cardiovascular disorder, which of course will, are also killers. But of course, takes a longer time, so we don't pay much attention to it. No? Unlike the COVID-19, which is very spectacular and so dramatic that we notice it and are afraid of it. Okay, we have deviated. Glucose in the blood is the major fuel source that is oxidized by our cells to give us ATP. Now, after a full meal, your glucose is converted to glycogen. Or if there's so much glucose, it's converted to triacyl glycerols, both of which are stored. Glycogen is an immediate source of glucose, no? So that glucose consuming organs or tissues can get it so easily. While some other tissues that prefer to have fatty acids, so they have a store of fatty acids in the form of triacyl glycerol. Okay. Now, glucose can be converted to compounds such as proteoglycans, glycoproteins, and glycolipids. So, when glucose enters the cells, it is right away phosphorylated so that it cannot go out anymore. We call that glucose trapping. We trap the glucose inside the cells. Now, for that glucose to come out, we have to remove the phosphate. But if a glucose has a phosphate, it cannot come out anymore. So that's why it's called glucose trapping. So glucose 6-phosphate is the pivotal compound for a number of metabolic pathways for glucose. Glucose 6-phosphate can be converted to UDP glucose, which is used for the synthesis of glycogen, or it is also used for compounds such as proteoglycans. Glucose 6-phosphate can also enter the pentose phosphate pathway or your hexose monophosphate shunt that produces NADPH for reductive biosynthesis. So it is used for synthesizing molecules, bigger molecules from small molecules. Example, fatty acids are formed from acetyl-CoA and they are put together in the presence of NADPH. Cholesterol as well comes from acetyl-CoA and they are put together in the presence again of NADPH. Okay? And the other product of your hexose monophosphate shunt is your ribose 5-phosphate for formation of nucleotides. Nucleotides are formed from ribose 5-phosphate, nitrogenous base, two kinds, purine and pyrimidine, and then phosphates. These are nucleotides. Nucleotides are put together to form nucleic acids. You have two types of nucleic acids, DNA and RNA. Okay? All right. So the major fate of your glucose 6-phosphate is to enter the pathway of glycolysis. Most tissues has the glycolytic pathway. And there's only one tissue that does not have it. A full glycolytic pathway. When I say full, it can be either aerobic or anaerobic. Now, one tissue has glycolysis, but it is only anaerobic. And that is your red blood cell, your RBC. No, so as I've said before, it is really paradoxical why it should go into anaerobic when in fact RBC carries oxygen. Yes, it carries oxygen, but it cannot use the oxygen because it does not have a mitochondrion. Mitochondrion are removed or other organelles of the RBC in the process of maturation are removed so that mature RBC does not have organelles. What replaces the organelles are molecules of hemoglobin. And that is its function, to carry hemoglobin. Principal function. Huh? Okay. So it is the glycolysis, which produces pyruvate and generates NADH and ATP. There are two sides of your glycolytic pathway where substrate level phosphorylation occurs. And there is one area or one step in which you produce an NADH. And we know that your NADH or a reduced NAD will be reoxidized, and it has been to be forwarded to the mitochondrion, specifically to the inner mitochondrial membrane, where you have your electron transport chain, in which you form ATP. For every molecule of NADH, you'll be able to gather 2.5 ATP. Okay? Now, fructose and galactose are converted to intermediates in the pathway by which glucose is metabolized. If it is not converted to glucose. Now, if it is converted to glucose, they start as glucose 6-phosphate. However, they have also different pathways for fructose and galactose, but its intermediates that will be formed will be also a part of the intermediates in glycolysis. Okay? We all know that glycogen is the storage form, the major storage form of carbohydrates in animals. The largest stores are found in the muscles and in the liver. Liver glycogen is used to maintain blood glucose when the blood glucose goes down, as in exercise, no, or fasting. Okay. Now, muscle glycogen is utilized to generate ATP for muscle contraction. For muscle contraction. It is not converted into free glucose to go into the blood, or meaning to increase the supply of glucose in the blood. Your muscle glycogen is used only by the muscle itself, because there is one lacking enzyme in the glycogenolysis of glycogen in the muscle, and that enzyme is your glucose 6-phosphatase. Okay, so the maintenance of the blood glucose is the major function of the liver, which produces glucose by the breakdown of glycogen or gluconeogenesis. And that is production of glucose from non-carbohydrate sources. Okay? Coffee to make me awake. All right. Oh, I did not mention about carbohydrate structure, no? Okay. So I mentioned already your carbohydrate as the definition, no? And then carbohydrates are compounds that usually contain an aldehyde or ketone group and a number of hydroxyl groups. We mentioned already the functional groups, no? Monosaccharides is the simplest carbohydrate. It can be of a D or L series. Okay. Now, what do you mean by stereoisomers? The simplest definition is that they are mirror images of each other. Mirror images. So when you look at yourself in the mirror, your right side, no, becomes the left side in the mirror. Diba? So that is when you look at yourself in the mirror. And it is the same thing with these chemicals or these compounds. Okay, stereoisomers have the same clinical formula and differ only in the special arrangement of their substituent groups. Substituent groups. Enantiomers. What do you mean by enantiomers? Enantiomers, they are stereoisomers that are mirror images of each other. So that is enantiomers. Now what are epimers? Epimers are also stereoisomers that differ in the position of the substituent group at only one carbon atom. Example, glucose and mannose are epimers in relation to carbon 2. So you will see that at carbon 2 of this, there is a difference in the position, no, of the OH and the hydrogen. So they only differ in that carbon, particular carbon. And glucose and galactose, they differ at carbon 4. Okay? They differ at carbon 4. All right. So they can easily be interconverted. If you have an enzyme, epimerase. Okay. Monosaccharides forms rings. Now we have seen the Fisher configuration, and that is the straight chain formula by Fisher. And we also have a ring form. So monosaccharides that have 5 carbon atoms forms a furan ring. Monosaccharides that have 6 carbon atoms forms a piran ring. So if a glucose is in a piran ring, so you call it glucopyranose. Piran for the ring. But if you just see glucose, it is in a straight chain formula. But when you say glucopyranose, then it is in a ring form. Or when you see ribose, usually it's 5 carbons, ribofuranose, a furan ring. Okay. The hydroxyl group, on the anomeric carbon may be either alpha or beta configuration in solution. No? Alpha or beta. When we say alpha, the hydroxyl group is where? Pointing upwards or downwards? Hala, nakalimtan na.
Downward.
Below.
And when it is beta, It is above.
Upward.
Okay. All right. So you place the ring like this, not like that. Place the ring like this. So it is up and this is down. So hydroxyl is up and that is hydroxyl down. Hydroxyl up, beta, hydroxyl down is alpha. No? Okay. What else? The monosaccharides are joined by a glycosidic linkage, no, an oxygen glycosidic bond or linkage to form disaccharides, oligosaccharides, and polysaccharides. You can also find glycosidic linkage in nucleotides, but it is not attached to oxygen. It is not an oxygen glycosidic bond. It is a nitrogen glycosidic bond. So oxygen glycosidic bond for carbohydrates, nitrogen glycosidic bonds for nucleotides. Now, your monosaccharides, or for that matter, the carbohydrate, contain phosphates, amino acids, or sulfate groups. No? Monosaccharides can be oxidized to corresponding acids. Example is glucose oxidized glucuronic acid, or reduced. So glucose reduced is known as sugar alcohol, or glucose, there's a term is sorbitol. No? Sorbitol. Fructose can become sorbitol, and galactose has a very nice name, dulcitol. Dulcitol or galactitol can be used. Galactitol or dulcitol. Okay. All right. What else? Now, when your monosaccharides or your glucose in a ring form, you can have a chair form or you can have a boat form. Huh? Okay. What else? Okay. Glycogen. Glycogen, a major storage of carbohydrates in animals, consists of a chain of alpha 1,4 linkage. So if your glucose are in a chain in an alpha 1,4 linkage, you have a straight chain. However, you can have an alpha 1,6 linkage, and in that case, you created a branch from the chain of alpha 1,4 linkages. Okay? Now, in glycogen synthesis, what is important is your UDP glucose, which will be acted upon by your glucose synthase to be added to a glycogen primer. You know what is a glycogen primer? A few glucose molecules attached to a protein known as glycogenin. Now, these few glucose molecules attached to glycogenin cannot be removed even in acute fasting. Now, the idea there is that your liver or your muscle still has this glycogen primer, so that when there is already glucose, or the animal is already eating, there's already food, then the glucose can be added to your glycogen primer so that you can store your extra glucose into glycogen in the liver or in the muscle. Now, glycogen degradation, or what you call glycogenolysis. The principal product is glucose 1-phosphate. And then, of course, glucose 1-phosphate becomes glucose 6-phosphate. And then glucose 6-phosphate is dephosphorylated by your glucose 6-phosphate phosphatase. Removes the phosphate. You have now a free glucose. And this free glucose augments or increases the amount of glucose. Now, that glycogenolysis, as we said, the major product is glucose 1-phosphate. But it can also produce free glucose through the action of your debranching enzyme. Still remember your debranching enzyme? Now, in glycogenolysis, glucose units are removed from the non-reducing ends of the glycogen chain by phosphorylase, which produces glucose 1-phosphate. Now, when you reach about 3 or 4, glucose left in a branch, there is a branching enzyme, which is glucose 4 transferase to the non-reducing end of another chain. So you have there a transfer. And what is left is just one glucose molecule in the branch, which is a 1,6 linkage. And this is acted upon by a debranching enzyme. And this time, free glucose is formed. Liver glycogen is used to maintain blood glucose. We mentioned that already. During fasting or exercise. Its breakdown is stimulated by glucagon and by epinephrine via a mechanism that involves cyclic AMP. Okay? Muscle glycogen is utilized to generate ATP for muscle contraction. Epinephrine again, via cyclic AMP, stimulates muscle glycogen breakdown. Okay? All right, let's take up glycolysis. Glycolysis is a pathway by which it oxidizes glucose and to produce pyruvate. And in producing pyruvate, it produces ATP through what you call a substrate level phosphorylation. Substrate level phosphorylation. Okay? And the product pyruvate in aerobic glycolysis is shunted to the mitochondrion. And as it enters the mitochondrion into the matrix, you have the enzyme pyruvate dehydrogenase complex, which converts your pyruvate into acetyl-CoA. And acetyl-CoA becomes now the substrate for your citric acid cycle. And you produce also ATP to substrate level phosphorylation, as well as reducing equivalents in ADH and FADH2, which is reoxidized in the electron transport chain, and you produces ATP. A molecule of NADH produces 2.5 molecules of ATP, and your molecule of FADH2 produces 1.5 molecules of ATP. Okay? All right. Now, I forgot to mention that the product of your glycolysis, you have pyruvate, you have ATP, it also produces NADH. And it's the same thing that NADH goes to your electron transport chain to produce ATP again, 2.5. Now remember, the NADH produced in the glycolysis is in the cytoplasm. The NADH produced by the citric acid cycle is inside the mitochondrion. Now, the NADH from the cytoplasm is brought to your electron transport chain to the inner mitochondrial membrane through a shuttle, which shuttles your reducing equivalents from the cytoplasm into the mitochondrion. So there is a shuttle for that, because NADH from your glycolysis is cytoplasmic, while the NADH in the mitochondrion is already inside the mitochondrion. So there's no need of a shuttle. Huh? Okay. All right. So what did I say? Okay. The NADH produced in the cytosol cannot directly enter your mitochondrion. So it has to be shuttled into your mitochondrion to generate ATP. Okay. Now, we know that your pyruvate that enters the mitochondrion acted upon by your pyruvate dehydrogenase complex can become acetyl-CoA. But you should also remember that the same pyruvate that enters the mitochondrion can be converted to oxaloacetate. And this conversion to oxaloacetate is in the process of gluconeogenesis. So which means that in the times of plenty, meaning glucose, the pyruvate is converted to acetyl-CoA. In the times of fasting, where glucose is low or deficient, the pyruvate that enters the mitochondrion is converted to oxaloacetate for the gluconeogenetic pathway. You get that? The same pyruvate, no? But in different situations, no, they form different compounds. In times of plenty, the pyruvate becomes acetyl-CoA. In times of fasting, the pyruvate becomes oxaloacetate. And we know that oxaloacetate is an intermediate in the Krebs cycle. And in gluconeogenesis, intermediates of your Krebs cycle forms part of the gluconeogenetic pathway. And that oxaloacetate, no, becomes malate, and malate exits out of the mitochondrion, and then it, through a carboxykinase, becomes phosphoenol pyruvate, and phosphoenol pyruvate now in the glycolytic pathway, it is a reversal, no, and it goes up and forms glucose. Of course, you have to have bypass enzymes. No? You have to have bypass enzymes. Okay. What else? Gluconeogenesis. Gluconeogenesis is the synthesis of glucose from compounds that are not carbohydrate in nature. It occurs mainly in the liver. In humans, the major precursor for gluconeogenesis are lactic acid or lactate, glucogenic amino acids. You still remember them? No? You have glycerol and the last three carbons of your fatty acid undergoing beta oxidation. And we are referring to propionyl-CoA. So propionyl-CoA can become So that's the only part of your fatty acid that can become glucose. And this does not occur in saturated fat, in even numbered fatty acids. It occurs only if the fatty acid undergoing beta oxidation is odd numbered. Now, in our body, our fatty acids synthesized by our body are even numbered. So where do we get this odd numbered or odd carbons, no, in your fatty acids? You get it from meat of ruminants, those that are eating grass. They produce this kind of fatty acid. So if we eat the meat of these ruminants, carabaos, cows, goats, sheep, so these are ruminants. And that is mostly our source of meat. And they call it red meat. So they can be form your odd numbered fatty acid. Now, since beta oxidation is removal of 2 carbons at a time, so if you are using an odd numbered fatty acid, you always end up with 3 carbons. And these 3 carbons are transformed right away into propionyl-CoA and enters the Krebs cycle at the intermediate succinyl-CoA. Becomes succinyl-CoA. So, since they are now intermediates of the Krebs cycle, they can be candidate for as precursor for forming glucose in your gluconeogenetic pathway. Now, you should also remember gluconeogenesis is not a simple reversal of glycolysis. There are 3 areas in your glycolytic pathway in which it is irreversible. At the level of hexokinase or glucokinase, at the level of phosphofructokinase, and at the level of pyruvate kinase. And I want you to remember what are the bypass enzymes utilized in these irreversible steps of your glycolytic pathway. So those are the only 3. The rest are reversible. That is why you can reverse. No? But you need to have bypass enzymes for this particular 3 irreversible steps. Okay? So, for pyruvate, because we said we have our what you call this? Our sources or precursors, no, of for the gluconeogenetic pathway are lactic acid, glucogenic amino acid, glycerol, and propionyl-CoA. Now, pyruvate carboxylase. Pyruvate carboxylase. Now, why are we talking about pyruvate? You should remember that lactic acid is pyruvate that is being reduced. Oh, being oxidized. And if you are going to reduce this, is become pyruvate. Correct? And your enzyme pyruvate carboxylase can convert that into oxaloacetate. So your lactate can become pyruvate. And pyruvate can become oxaloacetate. And oxaloacetate is a member or an intermediate in your Krebs cycle. So what happens now is that it can be used as a precursor for glucose formation. Okay? So oxaloacetate to malate, no? Malate exit, okay? And then becomes phosphoenol pyruvate through a carboxykinase. And then it goes up. And then you have reversal enzymes there for phosphofructokinase. You have fructose 1,6 biphosphatase. And then you have for your hexokinase or glucokinase reaction, you can have a bypass in which you have glucose 6 phosphatase so that you will have free glucose. No? All right. You can also get oxaloacetate from the carbon skeleton of the glucogenic amino acid aspartate. You remember? Aspartate is the carbon skeleton is oxaloacetate. So, which becomes malate and travels to the cytosol and is converted to phosphoenol pyruvate, no? And then it goes up in your glycolytic pathway. That enzyme, phosphoenol pyruvate carboxykinase, phosphoenol pyruvate carboxykinase converts oxaloacetate, no, to phosphoenol pyruvate. Phosphoenol pyruvate. Then phosphoenol pyruvate, as it goes up, it becomes fructose 1,6 biphosphate. And you have your fructose 1,6 biphosphatase, no, which is the bypass enzyme for phosphofructokinase. And then to glucose 6 phosphate, and you have your bypass glucose 6 phosphate phosphatase. Okay, gluconeogenesis occurs under conditions in which pyruvate dehydrogenase, pyruvate kinase, phosphofructokinase, glucokinase or hexokinase are relatively inactive. No? Relatively inactive because you have your bypass enzymes there. The low activity of these enzymes prevent futile cycles from occurring and ensures that overall pyruvate is converted to glucose. So the synthesis of one molecule of glucose from two molecules of pyruvate requires the expenditure of 6 molecules of ATP. See? Quite expensive. So where does this ATP come from? Because in gluconeogenesis, it is fasting. Diba? Or uncontrolled diabetes mellitus. No? So if it is from fasting, or there's no glucose, where does this ATP come from to form glucose in the process of gluconeogenesis? Where? Fatty acid beta oxidation. Oxidation of fatty acids. That is why fatty acid beta oxidation goes hand in hand with gluconeogenesis because gluconeogenesis derives the energy from the production of ATP by your beta oxidation of fatty acids. Okay. You have it. No? It's very good. You remember your beta oxidation yesterday. Okay. Now let's look at pentose phosphate pathway. Pentose phosphate pathway generates NADPH for what you call it, reductive biosynthesis and protection against oxidative damage. No? Oxidative damage. What particular oxidative damage? RBC hemolysis, red blood cell hemolysis. Because without NADPH, glutathione reductase cannot work. And it is your glutathione that prevents the damage by free radicals on the cellular membrane of your RBC. Now, in order to recycle the oxidized glutathione, it has to be reduced. And you have your NADPH as a source of your hydrogen. No? In order to reduce the oxidized glutathione. That's the most important function of your glutathione. Prevention of your damage to your cellular membrane. So much so that if there is something wrong that will result to the deficiency of the first two enzymes, of your hexose monophosphate, which reduces so much in ADPH production, then expect to have hemolytic anemia. Your red blood cell becomes very, very fragile. The membrane breaks easily as it passes through the graveyard. What is the organ graveyard of RBC?
Spleen.
Spleen. Now the spleen is like a filter. It has very small holes. And the red blood cell has to pass through those holes. So they squeeze inside. No, they squeeze through the hole, the small holes. So you need to have a strong membrane to do that. Now, if your membrane of the RBC is fragile, then it breaks. And that is the result of your hemolysis as it passes through the spleen. No? That's why it's called the graveyard. Because weakened or old age, senior citizen RBC cannot pass through the holes anymore without breaking. So it breaks. So it becomes the graveyard of old senescent RBC, or you call it senior citizen RBC. But healthy RBC, plus the support of your antioxidant glutathione, then it can easily pass through those holes, through those filters. So it's very easy for them to squeeze in without damaging the membrane. Okay? All right. In the irreversible oxidative steps, that's the first two reactions of your pentose phosphate pathway. In the irreversible oxidative steps or reactions of the pathway, 1 carbon of glucose-6-phosphate is released as carbon dioxide. NADPH is generated and ribulose 5-phosphate is produced. NADPH is used particularly for, we have seen it already, protection of your RBC membrane. It is also used for fatty acid synthesis and to reduce compounds, such as glutathione, as I've mentioned. Ribulose 5-phosphate provides ribose 5-phosphate for nucleotide biosynthesis, or generates pentose phosphates, which enters the non-oxidative portion of the pathway. The non-oxidative portion of the pathway is after the first two reactions. So meaning to say, in the third reaction and down. No? Because the irreversible steps or the oxidative phase is the first and second. Now, after that, you have reversible, non-oxidative reactions. The pentose phosphates produced from ribulose 5-phosphates may be converted to intermediates of glycolysis. So you can enter, this can enter into the glycolytic pathway. A series of transketolase and transaldolase transfers carbons from one compound to another. Transketolase which requires thiamine pyrophosphate. So thiamine, a vitamin B1 derivative, now becomes a coenzyme, thiamine pyrophosphate, TPP, abbreviated as TPP. Okay, transfers a 2-carbon unit. Then the aldolase transfers a 3-carbon unit. The net result of this reaction is that 2 molecules of cellulose 5-phosphate and 1 of ribose 5-phosphate, 2 molecules of fructose 6-phosphate, and 1 molecule of glyceraldehyde 3-phosphate are formed. So fructose 6-phosphate and glyceraldehyde 3-phosphate enters now your glycolytic pathway. They enter into the glycolytic pathway. Now, that is your pentose phosphate pathway, its importance. Okay, so what else do we have to talk about? The fructose and galactose metabolism. Although glucose is the most abundant monosaccharide derived from the diet, fructose and galactose are usually obtained also in quite a significant amount, mainly from sucrose and lactose. After fructose and galactose enters the cell, they are phosphorylated on carbon 1 and converted to intermediates in pathways of glucose metabolism. So they enter into the glucose metabolism, and that is your glycolytic pathway. The fate of group fructose and galactose is related to that of glucose. So they enter into the glycolytic pathway. Fructose is metabolized mainly in the liver, where it is converted to fructose 1-phosphate, cleaved to produce dihydroxyacetone phosphate and glyceraldehyde, which may be phosphorylated to glyceraldehyde 3-phosphate. These 2 triose phosphates are intermediates of your glycolysis. So they can enter into the glycolytic pathway. Fructose is metabolized. Fructose may be produced from sorbitol, which is generated from glucose, or fructose. What is significant of this sorbitol? Now, your eye lenses contains an enzyme, a reductase, which reduces glucose, and you form a sugar alcohol sorbitol. Now, fructose likewise can be reduced, and the resulting compound is also sorbitol. Excuse me. Which is also sorbitol. Okay. Now, sorbitol is especially in diabetics. The sorbitol in the lens becomes a nidus or a nest for the formation of cataract. So that's why diabetics type 2 usually develop diabetic cataract because of this. No? Galactose is phosphorylated at its carbon 1 and becomes galactose 1 phosphate, which reacts with UDP glucose. The products are glucose 1 phosphate and UDP galactose, which is epimerized through an epimerase to UDP glucose. The net result is that galactose is converted to glucose moieties of the UDP glucose and glucose 1 phosphate, intermediates in the pathways of glucose metabolism. UDP galactose may be also used in the synthesis of glycoproteins, glycolipids, proteoglycans. UDP galactose may react with glucose in mammary gland, active mammary gland, to form the milk sugar lactose. Now, galactose can also be reduced like your glucose and fructose to sugar alcohol. And the sugar alcohol of galactose is dulcitol or galactitol. So they are just the same. Galactitol or dulcitol. Huh? Tol. So otol, that is brother, no? Or sister. So if you have children, you have an otol, so yes, you can call him dulcitol. Or galactitol, brother of. It's just like the iris. Ever wondered why iris or names are have Mac, like McDonald, MacGyver? Those are Irish names. Mac means son of. So when you say McDonald, you are the son of Donald. Not necessarily Donald Trump. Okay. But we Filipinos, we have otol. No? Okay. So what do we have? More? I guess we have mentioned that, no? Hey, so our bimonthly is just carbohydrate and carbohydrate metabolism plus Dr. Barreto's lecture on plasma proteins. Okay? Don't you worry, the questions will be easy.
Thank you po.
Thank you, Doc.
Okay, thank you. Thank you also.
Thank you po, Doc.
Thank you, Doc.
Thank
Thank you po, Doc.